Okay, good morning, Professor Karaman. We are happy to meet you, even if virtually. Professor Karaman is a distinguished professor of pharmaceutical science of the College of Pharmacy at Al-Quds University in Jerusalem, Palestine. Together with his team, research new drugs applying computational methods. His research is also aimed at the synthesis of prodrugs to be used for the administration of certain drugs that have poor solubility in water or have a low bioavailability, as well as the synthesis of prodrugs to mask the bitter sensation of commonly used drugs. Today, Professor Karaman will hold a seminar entitled Dopamine, Gabapentin, and guaifenesin prodrugs. Please, you can start.
Thank you, Laura, for the introduction. Good morning, everybody. I will try to present now. Okay, you're in the screen. Okay. Enter screen. I'm trying to connect to the PowerPoint presentation. I should use what? The present now?
You must use in Flash. You have a Flash.
Flash, turn on.
Ne-
There is a key with present now. To
Yes,
click here
exactly. Presenta ora. Yes, exactly.
and a tab or a window or your entire screen. What to?
What you want. Well, you can use also the total screen.
Okay, I will try the entire screen. Okay, what's this now? Okay, share.
Okay, very well.
Okay, now I have to go to the file. Can you
Okay.
see the file?
Yeah, yeah,
Okay, now, right?
yeah, yeah. Very well.
Okay, good morning. My talk today is about dopamine, gabapentin, and guaifenesin prodrugs. Uh, Dopamine and gabapentin are drugs used for epilepsy and Parkinson disease, and guaifenesin is an expectorant used for adults and children. This picture contains some of my group, master students and PhD students that work in this project. First of all, I have to define what are prodrugs. Prodrugs are compounds which are inactive in vitro, and when converted in the body, they become active. Why we should use prodrugs? Prodrugs are used when the drug has acid sensitivity, poor membrane permeability, toxicity and side effects, bad taste, short duration of action, bad solubility, and inadequate stability. So these cases where we make from the drug prodrugs to improve these properties. Now, if you look at this figure, the drug itself cannot penetrate the membrane. So what we do, we just link the drug to a linker or promoiety, which makes the promoiety drug moiety both. When it's linked covalently to the drug, it allows it to penetrate the membrane and goes to the blood circulation. When it exposed to the blood circulation, it degrades to give the drug and the promoiety, and the promoiety should be non-toxic. This is the prodrug approach. which means we just change the physical properties of the drug in order to penetrate the membrane and to achieve the target in the blood circulation on the organs. As I said, prodrugs themselves are inactive, but when they convert in the body, they become active. Usually, most of the drugs are esters. When you make from the drug esters, we make it less polar, which allows penetrating the membrane. Now, the nature of the ester can be altered to vary the rate of hydrolysis, which means by changing the group of the ester, we can change the rate of the hydrolysis of the ester. We can utilize the similarity of the linker to biosynthesis building blocks, which are known in the body, to make it as a linker for the prodrugs. By doing so, we can enhance the permeation of the prodrugs through the membrane. Usually, a metabolic enzyme involved in the conversion of prodrugs to the active forms of the drugs. And if we know the metabolism of this enzyme, we can design a suitable prodrug. Not all prodrugs are activated by metabolic enzyme. Some of them, for example, photodynamic therapy, involves in the use of an external light source to activate prodrug. And some prodrugs are activated by chemical means, not by enzymes. When designing prodrugs, it's important to ensure that the prodrug is effectively converted to the active drug once it has been absorbed in the blood circulation. And we must be ensured that the linker or the promoter linked to the prodrug, to the drug, should be non-toxic. The prodrug to drug conversion can occur before absorption, before permeation, during absorption, or after absorption, or at a specific site in the body. In the ideal case, a prodrug is converted to the drug as soon as the desired goal for designing the prodrug has been achieved. Now, example of prodrugs, we have aspirin, as you know, is a prodrug of salicylic acid, the ester form of salicylic acid. Codeine is the prodrug of morphine. It metabolized in the body to give morphine. This is two examples of prodrugs. There are many examples also. Now there is an expression known as soft drug, and there is difference between soft drug and prodrug. Soft drugs are biologically active drugs. Prodrugs are inactive, but soft drugs are active drugs designed to have a predictable and controllable metabolism to non-toxic and inactive products after they have achieved their desired pharmacological effect. The opposite of prodrug. Prodrugs are inactive, and when they are exposed to the blood circulation, they become active. Soft drugs, they are active, and when exposed to blood circulation, they become inactive. The molecule could be deactivated and detoxified shortly after it has exerted its biological effect. This is the soft drug. The difference between prodrugs and soft drugs, the concepts of prodrugs and soft drugs are opposite as follows. A prodrug is an inactive compound that requires a metabolic conversion to the active form. A soft drug is pharmacologically active and uses metabolism as a means of promoting excretion. Now I'm going to talk about the three drugs that I introduced in the beginning. And I am going to talk about the disease that they treat. Parkinson's disease, which dopamine treat. According to the Parkinson's Disease Foundation, It affects about 1 million people in the United States and more than 10 million in the worldwide. The causes of Parkinson's disease, the exact cause of Parkinson's disease is still unknown, but they hypothesized that multiple factors acting together to make this disease, including aging, genetic and environmental exposure factors. A number of environmental factors have been associated with an increased risk of Parkinson's disease, including pesticide exposure, living in industrialized country, and drinking well water. These are the environmental risk factors for Parkinson's disease. Parkinson's disease is considered as non-genetic disorder. However, about 15% of the people that they have this disease have a first degree relative to who has the disease, which gives indication that it might be genetic. They found that mutation in specific genes can cause Parkinson's disease. These genes are, as shown here, SNCA, PRKN, LRRK2, PTEN, PINK1, and else. Dopamine for the treatment of Parkinson's disease. This is the chemical structure of dopamine. It's an amine. Parkinson patients have insufficient dopamine in specific regions of the brain, which makes the disease. They don't have enough dopamine in the brain. So attempts have been made to replenish the deficiency of the dopamine. They have to give the body dopamine, which should be in the brain. Dopamine itself does not cross blood-brain barrier because at pH 7.4, the amine becomes ionized and it inhibits its permeation to the membrane, the blood-brain barrier. The dopamine precursor L-dopa is actively transported into the CNS, which means the precursor, it penetrates the barrier. However, the dopamine itself does not penetrate the barrier. This is the structure of levodopa. The bioavailability of L-dopa is less than 10%, and only 10% of the 10%, about 1%, enters the brain. Large doses of levodopa are required because much of the drug is decarboxylated to the dopamine in the periphery. So, because we have these enzymes that can decarboxylate the L-dopa in the periphery, we need large doses of L-dopa. To minimize the conversion to dopamine outside the central nervous system, usually L-dopa is co-administered with amino acid decarboxylase like carbidopa and benzoylide. These two drugs, carbidopa and the other one here. And in spite of this, we still have side effects like dyskinesia, on-off phenomenon, and end of dose deterioration still remain. Levodopa provides the greatest anti-Parkinson benefit for motor signs and symptoms with the fewest adverse effects in the short term. However, long-term treatment leads to involuntary movement and response fluctuation, which add to the complexity of later disease management, which means we still have side effects if we use large amount of levodopa. In addition, preclinical evidence suggests that levodopa is toxic to dopaminergic neuron. Once flocculation and dyskinesia become problematic, they are difficult to issue. Still, we have problems with giving levodopa as a replacement of dopamine. So we should think about alternative of L-dopa. Also, this is about gabapentin, which treat neuropathic pain. What's neuropathic pain? Is triggered by damage of the somatosensory nerve system, affecting both its function and structure, and leading to sudden onset of pain. It's commonly described as pricking, tingling, burning, coldness, itching, or stabbing. It's estimated to distress millions of people worldwide and also has a significant effect on health-related quality of life and is associated with high economic burden for the individual and society. Gabapentin and its prodrug pregabalin have been shown to be effective in many neuropathic pain conditions. However, post-marketing has noted an increasing number of reports of heart failure when giving these drugs. Also, gabapentin is used in epilepsy. Epilepsy is a common chronic neurological and heterogeneous collection of disorders affecting about 65 million of people worldwide. It is characterized by repeated incidents of involuntary movement and unusual sensation accompanied by loss of awareness of the resulting recurrent seizure can be classified into either focal, including only part of the body, or generalized involving entire body, which constitutes many types such as absence, atonic, myoclonic, and tonic-clonic seizures. There is evidence that gabapentin has efficacy as monotherapy in adults and adolescents with newly recognized partial or mixed seizure disease, which means that gabapentin is effective in treating these cases. Also, the gabapentin is used in this syndrome, Brushless Leg Syndrome, which is a common neurological disease characterized by an urgent need to move the legs to alleviate unpleasant, not usually painful, and uncomfortable sensations. These symptoms appear during the night or on rest, particularly when lying or sitting. It is divided into idiopathic and symptomatic forms of RLS, with the symptomatic form being associated with, for example, Parkinson's disease, iron deficiency, uremia, pregnancy, or chronic renal failure. Pregabalin, the prodrug of gabapentin, and gabapentin were recommended for initial RLS management. They are effective. Now, this is the structure of gabapentin. As you can see, it's like of amino acid. It's resembling GABA. GABA is an inhibitory neurotransmitter that was shown to have a role in managing seizure states. It's sweeter ion. It has 2 pKa, about 3 plus and 10 plus, and it's not metabolized by human. It's excreted renally without change at all. It has short T-half value and non-linear pharmacokinetic, which limits its clinical effectiveness. The bioavailability of gabapentin differs inversely with dose, which means once we increase the dose, the bioavailability decreases. Additionally, the absorption of gabapentin varies from patient to patient. It was approved in 1993 for anti-epileptic medicine. Now it has the approval for the treatment of several types of neuropathic pain, epilepsy, and RLS. It is well tolerated, but can cause dose-related dizziness and sedation. As a consequence of the pharmacokinetic deficiency of gabapentin, the prediction of its dose necessary to achieve an optimal therapeutic effect in a given patient is often difficult, and the desired treatment response may not be achieved because of the absorption phenomena that it has. In addition, the short T-half value of gabapentin represents another limitation of its pharmacokinetics that initiate frequent dosing, which is a cause of non-compliance in epileptic patients and missed doses which can reduce clinical effectiveness. Now we have, because the low bioavailability of gabapentin and low bioavailability of dopamine and all the side effects accompanied with the use of these two drugs, we have designed the dopamine and gabapentin products. The main goal of this research was to make products of dopamine and gabapentin that have the potential of higher bioavailability and have the ability to be hydrolyzed chemically without the use of enzyme, chemically, just chemical reaction, to give the parent drugs without the need of enzyme. Please note that the prodrug chemical strategy requiring enzyme catalysis, usually prodrugs needs enzymes, but in this approach we don't use enzymes. When we use enzyme, there are many disadvantages related to many intrinsic and extrinsic factors that can alter the catalysis process, such as unpredictable clinical effects and safety of the most prodrug activating enzyme owing to genetic polymorphism, age-related physiological change, or drug interaction. Which means by changing the approach, we can eliminate these factors that affect the bioavailability and the conversion of the drug to drug. We have explored a number of intermolecular processes to gain insight into enzyme catalysis toward the development of prodrug linker with improved bioavailability over existing medication using ab initio and DFT methods. First of all, what we have done is using computational chemistry to develop a method that can be used to make prodrugs. So we studied intermolecular process to see what linker to be used. We found that by using different linkers, we can achieve different rates, depending on the nature of the linker and the nature of the intermolecular process. For example, we studied the proton transfer reaction in some of chem's acid amide derivatives by using enzyme models as potential linkers to be linked to amine drugs like dopamine and gabapentin. The computational calculation using DFT method reveals a linear correlation between the calculated and the experimental value. What we have done is we took experimental experiments, and we calculated the rate, and we compared the rate, the calculated ones to the experimental ones, and we found a linear correlation between them. And by using this correlation, we can design another linker or different products. Based on this calculation, several dopamine and gabapentin prodrugs were designed, and their cleavage rates to their parent drugs were found to be largely dependent on the nature of the linker attached to the drug. Here, the synthesis of dopamine prodrugs. We have the dopamine. We use first step with sodium hydride. We just, after that, we condense that with the anhydride. And the anhydride, here should be oxygen here. Here oxygen and here oxygen. Plasmid anhydride, which the amine attack the carbonyl and give the prodrug here and here the prodrug. It's very simple synthesis. And the characterization of the prodrug by NMR, HNMR, C13 NMR, and LCMS after chromatographic separation. And we have done kinetics, in vitro kinetics for these dopamine prodrugs in different media, 0.1 normal HCl mimic the stomach. pH 2.2, the middle of the stomach. pH 5.5 is the beginning of the intestine, and pH 7.4 blood circulation. And we found the rate in each medium. It was noticed that in acidic medium it has the highest rate, and in basic medium or neutral medium it has a moderate rate. And we have done in silico admit prediction to see if these prodrugs can penetrate the brain-blood barrier. And from this table, we can find that these prodrugs are indeed can penetrate the barrier. In silico pharmacokinetic properties and toxicity were predicted for the two dopamine prodrugs, and results of the pharmacokinetic and toxicity prediction reveals that no prodrug has a high risk of toxicity, and both prodrugs showed good pharmacokinetic properties. In addition to the in silico, we did actually did toxicity in rats, and we found that these dopamine they have LD50, high LD50, which means they are not toxic. Here are some parameters of the in silico ADME. You can see we can calculate the oral bioavailability about 30%, and we can predict the transport, which is passive, for dopamine prodrug. And you can see the log BP is minus half. If it's above minus 1, it can penetrate the barrier. If it's less than minus 1, it has less potential to penetrate. We also investigated these prodrugs in the plasma and brain of mouse. This is the prodrug that we tested. This is the dopamine, and this is gives you the dopamine in brain when it gives dopamine alone and after prodrug administration. And we can see that the dopamine prodrug penetrates the brain, and you can see here it has about 2-3 hours. You can find it in the brain. In plasma, the dopamine, you can find dopamine in plasma. However, when you give the dopamine prodrug, there is no prodrug in the plasma, which is very good, because when dopamine exists in the plasma, it can make side effects, which is not good, hypotension and other side effects. If you give the prodrug, there is no dopamine in the blood, and which eliminate the side effects by administrating prodrug. Here is the prodrug in the plasma. You can see the prodrug in plasma and in brain, evidence that it enters the brain. About the gabapentin, we also the same method was used. Instead of sodium hydride, here we used sodium hydroxide and condensation with the amine, and give the gabapentin prodrug here and here, different prodrugs. And we characterize these prodrugs by NMR and LCMS, also FTIR and others. And we did in vitro kinetics for these prodrugs. As you can see, we have the same picture as for dopamine. In acidic medium, it has the highest rate, and in normal pH, neutral pH, the rate is less than acidic pH. And here we did also in silico prediction for the penetration, and we got log P, which is more than -1, and which means that this gabapentin prodrugs have the potential to penetrate the barrier. BBB permeability software predicted that gabapentin products that we made have the capability to test the barrier. Abraham et al. Suggested that compounds with log PP less than -1 will not cross through the barrier, while compounds with log PP equal to or more than -1 are predicted to enter to CNS. All the synthetic gabapentin prodrugs were predicted to have log PP more than -1, which means they are good. And here some figure of the in silico prediction. And we still have to give these to rats or mouse, mice, to see the actual penetration in vivo. Now, bitter guaifenesin prodrugs. This is guaifenesin, is an expectorant. It has a bitter taste, very strong bitter taste. Because of that, many children do not take this medicine because of the bitter taste. Now the idea was to make bitterless product for guaifenesin and to link guaifenesin to a linker, which upon exposure to the stomach, it degrades to give guaifenesin. And the hypothesis was the product should be bitterless because we just masked these hydroxyl, which predicted to make bonds with the bitter taste receptors on the tongue. Why we thought that? Because before that, we made that because we have shown that paracetamol prodrugs, paracetamol is a bitter drug, and when we masked this hydroxyl group with a linker, we found that this prodrug is bitterless, does not have a bitter taste, such as phenacetin, which does not have bitter taste also, because the hydroxyl, the phenyl group, is blocked by methylene. Now, masking the bitter taste of guaifenesin using product approach, we designed inside, also we used computational methods to design these prodrugs. We characterized the prodrugs after we made them. We performed kinetics for these, and we made tests for binding to receptor number 14. As you know, we have about 25 receptors for bitter taste, and receptor 14 is the major receptor that we know in the cell. Now, we also made bitterless antibacterial prodrugs. This is amoxicillin, this is cephalexin. They are bitter, they have bitter taste. When we just block the amino group here with this linker, we found that these prodrugs do not have bitter taste. Now, this examples how we do calculation by DFT calculation. You can predict the energies, the global minimum transient state for the reaction of the structure, all of these diameters. And here, table gives you strain energy, activation energy, and distances. Here we made the mono and di because you have two hydroxyl in the guaifenesin. We made the mono and diester of guaifenesin. And we characterize these esters, mono and di, by LCMS and NMR, HNMR, CNMR, C13NMR, FTIR. This characterization of some of these. And we did in vitro kinetic study for these products, and we found that all of them are stable in neutral pH, over they degrade in acidic pH, which this is what we want. We want it to reach the stomach and to be converted in the stomach directly to give the parent drug, which is very good because in neutral pH it's stable, and acidic medium it degrades to give the drug. And we did the bitter taste binding test, and we found that the diester do not have bitter taste, whereas the mono have moderate bitter taste, less bitter than the drug, but they are more bitter than the diester. Diester do not have bitterness at all. Summary: we have explored all possible mechanisms for the proposed enzyme model intramolecular processes. We utilize the computational method to make new important prodrugs for drugs that suffer low bioavailability and have bitter sensation. We studied the toxicity, pharmacokinetics, and in vivo of some of these prodrugs. And we found that the dopamine prodrugs, they do penetrate the blood-brain barrier. And we found that guaifenesin prodrugs do not have bitter sensation. And thank you for all. It was a pleasure sharing with you our own scientific experience. Thank you. I cannot hear the voice.
Hello, Rafik.
Hello. Hi, Sabino. How are you?
Hi. Fine.
How are you?
Thank you very much for your presentation.
Thank you, Sabino. Nice to see you.
Oh, thank you. Nice to see you too. Okay. Where are you at home?
I'm still, I'm at home now, but I am still at Al-Quds University.
Uh-huh.
Where to go?
What is the situation there?
It's a little bit calm, no problems. Meanwhile, but who knows?
The pandemic?
No, we have a good situation. We don't have a lot of cases. We have about 500 cases every day.
Okay. So I am glad to see also Amin.
Yeah, I
Amin
see.
is there.
Now, I mean,
Hello.
I have to tell you that Dunya is now a professor, assistant professor at Al-Quds University.
It's a
very well.
good-
Hi,
good
professors.
news.
Hi, Amin.
How are you?
Fine. And you?
Me too. I am fine. What's the situation now with the coronavirus in Italy?
We are starting with the third.
We're starting before you.
The third round of vaccine.
Yeah, we're starting to general life before you now.
Oh yes, I know.
We are now going to university, to markets. It's usually to go everywhere without masks, without any protection. Maybe because we are taking the vaccine.
Yeah. We too. Okay. Okay.
Nice presentation from Professor Rafik, and this is a nice meeting with you and the professors Laura Schirano and the others. So thank you for this meeting.
Yeah, but I wait your future presentation.
I want to talk about the prodrugs. So me and Professor Rafik, we are one now.
Ok. Ragazzi, avete domande? Have you question? Siete tutti senza parole? Any question? Nessuna domanda. Tutto molto chiaro. Ok. I stop.
I have a question for
Yes, Amin.
Rafik and for Amin.
Yeah.
Because yes, you know that I was involved in your research in the past. So I am asking for a new research, a new active research for the next future. What kind of drugs you think it's possible to modify to eliminate the problems that Rafik highlights?
So which kind of research you want in prodrug, in water treatment, in what kind you want?
Naturally, the first one is in prodrugs.
Yeah, industrial chemistry.
What do you think for the next future?
The next future of prodrugs, you know, Professor, and all that drugs have a lot of problems, so it's need some modification, like enhance the permeability of the drugs, enhance the taste of the drugs, enhance everything for the drugs. So all the drugs need some modification to be good for administration, or to enhance its specification for children, for all people. So I think the future of the prodrugs, or the prodrugs, is the future of the pharmaceutical industry.
Ah, and what about the possibility to collaborate on a new medicine for COVID?
Everything need money.
Yeah, this is Rafik have a lot of money.
You know, I don't have.
He can contribute.
I wish I can.
Yeah,
I
but
wish I can.
you can present a research project to USA.
But you know, I am not working with COVID. I don't know this kind.
Yeah.
It's not my expertise. No, you need somebody who know this field.
Yeah, it's a big problem.
You know, I
because-
am an old man now. I cannot change my subject. You know,
Yeah.
but I mean, he's still young. He can.
Rafik, I am
pursue this kind
retired,
of research.
so I can understand you.
Okay. Okay.
So, thank you very much again.
Thank you, Sabino and Laura. I wish I can see you in the near future.
Okay.
you.
Yes.
I mean, when you can come here
Me or Professor Rafik?
with Rafik.
With Rafik.
Oh.
I think the situation in Italy, it's not constant. So the airports, it's not open all the day. Also, us to cross to Jordan, it's not easily because we are talking about from the state to another state. So we need a lot of tests for corona, a lot of papers, documents to pass through to Jordan. Also, if there is any invitation for conference, for something like that, offline conference, not online conference, face-to-face conference, we can manage to visit to Italy.
Yeah,
Okay.
I think that in the next year we can organize something.
Okay. Waiting for the next year.
Okay. Bye-bye.
If you invite me, I can come.
Okay.
Yes,
naturally.
the invitation for me and for Professor Rafik.
Yeah.
If you
Not for
invite
me
me,
only.
Sabino, if you invite me to 2 or 3 months, I can come.
Okay. I
Okay.
hope to be able.
Yeah.
Okay. Ciao.
Okay. Bye. Thank you.
Bye.
Thank you so much. Thank
Bye.
you.
Thank you, professors. Ciao.
Ok ragazzi,